For this video, we're going to start getting into water transport. So for now, we're just going to kind of talk about the physics of how water moves and why it moves. And in later videos, we'll talk about how it kind of moves through different parts of the plant. So this should be some review, but we need to go over diffusion and osmosis. So remember, diffusion is just a movement of solutes from high to low concentrations. Like if we look in this picture here, this is a cell membrane right here, so the lipid bilayer. And on the outside, the extracellular piece here, we have a bunch of solutes. Now, notice they're in really high concentration relative to the inside here. And the inside has very low concentration, 0 at this point. If this membrane is permeable, meaning those solutes can move through that membrane, then they're going to diffuse from high to low concentrations. And this occurs just by random movement. So if we go over in time one little segment there, you can see some of these solutes are now starting to diffuse from high to low concentrations. And then go over a little bit more, and you can see now they're at equal concentrations, 50-50 on each side. And this is the same idea as if you sprayed perfume in the corner of the room. The perfume would, through random movement, diffuse throughout the entire room until it's in equal abundance. So that's diffusion. And certain things can move through the membrane like that, particularly small molecules that aren't non-polar molecules can move through it relatively easily. The larger and the more polarized they are, the more inhibited they are of moving through the cell membrane. Now, if we look down here, and we'll look at this on the next slide as well with the plant cell, but you can see there's a cell here in the middle, and they're inside of solutions. And the solutions on the outside of the cell change in the concentration of solutes, so the number of solutes that they have in each image. So if we look here, this is a hypertonic solution, meaning the outside solution has a higher osmolarity than the inside of the cell. Osmolarity being the number of solutes. If the solutes are in really high concentration and they can't move through the membrane's not permeable to them, then water will move, and that's osmosis. Osmosis is the movement of the solvent, usually water. So in this one, if the solute concentration is really high outside and relatively low inside, water will move down its concentration gradient. So if you look inside the cell, it has 20% solutes and 80% water. The outside has 60 or 40% solutes and 60% water. 80% water to 60% water means water will want to move out of the cell. It'll move down its concentration gradient, just like a solute would. And so as a result of that, the amount of liquid in the cell becomes less and the cell shrivels up. Now over here in the isotonic solution, they have the same concentrations inside and outside the cell. And so the cell doesn't change. Water moves in and out of the cell at the same rate. Now in hypotonic solution, the cell has a higher solute concentration than the outside. Meaning there's now 80% water in the cell and 90% of water outside the cell. In that case, water is going to want to move from the solution, from the outside, into the cell. And so it'll move into the cell here. And in some cells, this can cause it to blow up. It'll burst. In plant cells, they're protected because they have a cell wall. So if we look over here, they're lined up the same way, hyper, iso, and hypo. Oops. Hypertonic, remember the solution is higher outside the cell, and so water moves from in the cell to the outside. And you can see here the central vacuole, the part that's going to hold the water in the cell, shrinks. Now, we still have the cell wall. The cell wall is nice and rigid, so it doesn't really affect the cell too much, although it can cause wilting and different things like that, depending on the conditions. Isotonic, water moves inside and outside the cell at the same rate. And then hypotonic, a bunch of water flows into the central vacuole. The central vacuole expands and pushes against the cell wall. We call that turgor pressure. So the pressure of the water forcing the cell membrane out into the cell wall, and it creates a rigidity to the cell. Most plants want to be in a turgid state. They want to have that ability to maintain that rigid structure so they don't wilt. A plant in a hypertonic solution will often wilt. Now, we're going to talk about water potential and why and when water moves, but keep in mind that in a hypotonic state, water moves into the cell and creates turgor pressure. And turgor pressure is that pushing of the water against the cell wall, creating that rigidity. Now, there's three main different ways that solutes move across the membrane. One of them is just simple diffusion. So what we talked about here, right? If the membrane is permeable to that solute, then they can just move across the membrane. No big deal at all. But if, say, this membrane here is not permeable to these solutes, then they can't move across the membrane. They're stuck on the outside. But what if the cell wants those solutes inside the cell? So it does want them to come in, but they can't because they're not permeable. In that case, the membrane will create proteins that will act as channels for those solutes to move into the cell. So here's an example of a protein channel. Now these ones they're specific to different solutes, but so this one's specific to these green hexagonal ones. And if it's a higher concentration out here, they're going to diffuse through, and they're going to diffuse through that protein channel rather than going through the membrane. The benefits of this is that you can bypass the permeability of the membrane. You can regulate what can come in and out of the cell. The downside to it is that in simple diffusion, all of the solutes can diffuse anywhere in the membrane. And here they have to go through a protein channel, which means it is a slower form of diffusion because these solutes kind of have to randomly encounter that entrance and then move through it, versus just being able to move to the membrane kind of freely. Still, though, it doesn't require any energy, and it only goes from high to low concentration. It's just like simple diffusion in every way, except that it's not permeable to them. The membrane is not permeable to them, so they move through a protein. Then we have active transport. So active transport requires energy, and it's usually going against the concentration gradient. So simple and facilitated diffusion is going from high concentrations to low, right? And that's free. You don't need to add any energy to the system for it to do that. But if you want to go from low concentration to high, you have to put in some energy. One way to think about it is if there's a rock on top of the mountain, it can just fall down the mountain. It can just roll down. It doesn't require any energy. But if you want to move it back up the mountain, you have to physically push it back up, and that takes energy from you. Active transport is pushing the rock back up the mountain. So here we have some different membranes here, and we have potassium ions and sodium ions. And if we look at the concentrations over here, the sodium ions, or potassium ions, are really high inside the cell, and the sodium ions are really high outside the cell, according to this little chart here, this color diagram. Now, you can see these different protein channels here, and what this will do, this is a sodium channel, it will load sodiums into here, and remember it's already high on this side of the cell, and it wants to move sodiums outside the cell. It'll load sodiums into this protein, and then add ATP. Remember, ATP is adenosine triphosphate. It's the currency of the cell. It's how we get energy from the metabolic pathways like cellular respiration, and then use that energy for physiological processes like transport of ions. So this ATP molecule will go over to the sodium. You can see that here in this next diagram. The terminal phosphate, the last phosphate, will break off, and that energy from that bond will go to the protein channel. So you can see that little energy here from this phosphate breaks, and it goes to the protein channel here, and that triggers the protein to change shape. So it goes from this to this. And as a result, the sodium ions that were sitting on the inside here are now on the outside. And so they can move from what's a high concentration or a low concentration here to a high concentration. And so it's moving the opposite way of simple diffusion. The same thing happened for potassium here. Potassium's high on the outside, and so it's going to load the concentration, load them into here, pay the ATP fee, and then move them inside the cell. So you have simple diffusion, diffuse to the membrane from high to low, facilitated diffusion, diffuse through a protein channel from high to low, and active transport, move from low to high through a pump that requires energy. And in some of the things we're going to talk about this week, we're going to look at all three different types of transport and when they're important. Okay, so remember, for this video, we're focusing on how water moves. And so most of the video, we're going to be talking about water potential. Now, water potential is kind of a weird idea, but the simplest way to describe it, it's the potential energy of a system relative to pure water. So what that means is potential, there's different types of energy. Mostly potential and kinetic is how we kind of describe it. Kinetic energy is like when you're moving something, how much energy are you going to put into something as you're moving? So it's the act of actually exerting that force. Potential energy is the force that you could potentially exert. For example, if you take a rock and you lift it a foot off the ground, if it drops, it has energy that it can damage the ground with, right? And if you move it 5 feet off the ground, it has more potential energy and more likelihood of damaging the ground beneath it. So that potential energy is then just the energy that exists in the system if something were to happen, right? It could potentially use that energy. And so water potential acts the same way. It's a little bit more convoluted, and I'm not going to ask you to describe it in too much detail, other than knowing that definition, that water potential is the potential energy of the system. Now, we're going to be looking at a lot of formulas here, and they look complicated because they use a lot of fancy symbols, like this Greek symbol psi, which looks like a trident. It's really just addition and then some multiplication. And so it's not too complex. And I encourage you to not get bogged down in the symbols. I use them here because this is what you will see in all of the textbooks and anything that you look at regarding this material. They're going to use these symbols. And so I'm going to use the symbols so that way you're familiar with them. But basically what this is saying is that the total water potential of any given system, be it a cell or the soil or the air, whatever it is that you're talking about, is equal to the solute potential. So the potential driven by the solutes, the pressure. So like say like turgor pressure, that puts pressure on the inside of the cell. That potential, gravity. So as you go up higher, right, gravity is going to pull you back down. So the potential added by gravity. And then also what we call matric potential, the potential of it bonding to other things inside the cell that's going to kind of slow it down. And we're going to go through each one of these. Now, for each of these, they're going to be, actually, the water potential total, I sure circled all of them there. They're going to be measured in millipascals, so MPAs. It's just a unit of basically pressure, of potential. And so you're going to see MPAs a lot here, and that's just the amount of pressure that's being attributed to each one of these. So when we're talking about water potential, water will only move from one system to the next if the water potential total changes. So in general, what will happen is that, say you have two systems, like two adjacent cells, and the water potential of this one is, say, like -1, and the water potential of this one is -3, water will move from this cell to this one because it's going to go from the high to low pressure or low potential, right? So water is going to move from high to low water potentials. And we're often in these ones going to be dealing with negative numbers. So it's kind of easy to confuse a little bit, but remember, -1 is greater than -3. And so it'll move from -1 to -3. Now, changing one of these individual components doesn't necessarily change the total water potential, but it normally will. So for example, if this one was 1, water potential for pressure potential, and these ones were 0, and this one was 3, changing this one to 2 would mean that the sum of these is now less, and the total water potential has changed, right? And so it often will result in a change in water potential total, but sometimes these ones can act in concert and they can change in opposite directions, and therefore the total potential might not change, but it normally will. Here's an example down here. So we will describe, if you're talking about the type of potential, it'll be a subscript, so like S, P, G, and M, so solutes, pressure, gravity, and matric. If you're talking about the system, like the root or the soil or the cell, you put it as a superscript. So water potential of the root, water potential of the soil. And in these cases, they're talking about the total water potential. So water potential of the root is 0.3 millipascals. And the solute potential, or the total potential of the soil is -0.1 millipascals. -1 MPa is less or greater than -0.3 MPa. So water will move from the soil into the roots because this is greater than this one. And so it'll move to the lower water potential. Okay, so now we're going to move through each type of potential that's part of that overall equation one by one. Some of them we're going to spend a lot of time on, and other ones it's going to be mostly negligible, and it'll be just one quick slide. Solute potential is a big one, so we're going to spend quite a bit of time on this. The other way of thinking about solute potential is through osmotic potential. Now remember, when you talk about hypertonicity and hypotonicity, we use the word osmolarity. Is the osmolarity higher inside or outside the cell? That means the solute concentrations. And so solute potential is directly related to the solute concentrations and is therefore equivalent to osmotic potential. So the osmolarity, the potential driven by the osmolarity of the system. Now, we've talked about the unique properties of water previously. And water, we know, is the universal solvent. And part of that is because it's a polar molecule. The oxygen on the molecule is negative and the hydrogens are positive because oxygen tends to pull the electrons towards that side. So if we look at a water molecule here, you can see the oxygen here is negative and each hydrogen is positive. That makes water really good at breaking particles apart. And so say we have salt, sodium chloride, NaCl, because the sodium is positive, the oxygens will bind to it. Because the chlorine is negative, the hydrogens will bind to it. And as a result, water can kind of weasel its way into molecules and rip them apart, right? That's important because when we're talking about solute potential, NaCl would be one molecule. But when you break it apart, it's now two molecules. And so later on, you're going to hear us call that the ionization constant. How many particles do the solutes break into? Now, if it's a molecule like glucose or fat or something like that, or sucrose, which is a common one we talk about, water won't break it apart, and so it'll stay as one, right? Now, what's important here is remember, water potential is defined as the potential energy of the system, of that water, well as pure water. The number of bonds that are forming between water molecules and other things lower the potential energy of the system. So when these hydrogen bonds form between water and other things, other solutes, the overall energy of the system goes down. So the amount of potential energy that exists, some is being used up in those bonds. So the potential energy goes down in those systems. So the more bonds, the lower the potential energy. So more solutes means lower solute potential. Solute potential equals 0 in pure water because there's nothing else in the water for it to bind to. In plant cells, solute potential is always less than 0, and it cannot be positive. So solute potential is either 0 in pure water or less than that. Okay, so remember, here's our equation. So water potential, the total water potential equals solute potential plus pressure potential plus gravity plus metric, right? So adding solutes lowers solute potential because it's taking up more of those bonds. So more of that potential energy is decreasing. And the total water potential here will go down as well, assuming nothing else accounts for it, and normally it won't there. So let's look at this example. Here's pure water. Here's a permeable membrane where solutes cannot move, but water can. So it's semi-permeable. Water can move, but solutes cannot. You have this U-shaped tube. If you were to put pure water in there, the water would be level. It would immediately level out. Now, if you add a bunch of solutes here to the right side, right, that means the solute or the solute potential on this side becomes less. And if solute potential becomes less, total water potential becomes less, right? And water will move from high water potential to low water potential. So the water potential on this side is now higher than it is on this side. And so water will move to the right. So water is going to shift from the left side to the right side until those water potentials are equal. Now, plants are able to manipulate solute concentrations in their cells. Think about the diffusion, simple diffusion, facilitated diffusion, and active transport that we talked about earlier. If plants can use those to increase or decrease the solute potential or the number of solutes, then they can impact the solute potential, right? If you take solutes out, your solute potential is going to go up, and if you put solutes in, your solute potential is going to go down. So let's say you have a plant that has roots that are in the soil, and it wants to draw water in. But maybe the water potential of the soil and the root are the same. So in that case, water wouldn't move into the plant. But say the plant, for whatever reason at the time, needs some water, it could pump solutes into the roots, and that root would then have a lower water potential, and water would move from the soil into the root, right? So it's kind of like creating a hypotonic condition. You're pumping in a bunch of solutes into the cell, which makes the cell hypotonic relative to the soil, and water moves into the cell. So plants can pump in solutes to modify their solute potential and draw in or stop drawing in water. Now, solute potential, there's a calculation need for calculating solute potential. And that's because you often are given the molarity of the solutes or the cell and not the actual solute potential. So to calculate solute potential, which is the symbol here, you use this formula, negative I times C times R times T. Now, it's not a very complicated formula, but you do know how to use it. And on exams and quizzes, I will give you the equation. So in this case, I is the ionization constant, just like what we talked about a little bit ago. If you drop in salt, NaCl, sodium chloride, into water, water will split it into Na and Cl. So two different molecules. In that case, it has an ionization constant of 2 because it breaks into two parts. If you drop sucrose into water, it's not going to dissolve, it's not going to break down or dissociate. Therefore, it has an ionization constant of 1. It stays the same. And if something was to break into 3 parts, you would have the same response. You'd have an ionization constant of 3. Now, I don't expect you to have taken chemistry before, so I'm not going to require that you know what it breaks into. I will tell you what it will do. I'll say either assume that it's an ionization constant of 1, or I will say sodium chloride broke into sodium and chloride. And so that way you know there's 2 pieces, right? So you just have to be able to read the problem and understand that there are multiple pieces, 1, 2, or 3, whatever it might be. You don't have to understand if I'm not going to give you a molecule and hope and you understand what it will do. You're not required to do that part. Now, C is the molarity of the solution. And so if you haven't taken chemistry yet, that might sound kind of weird. But because solutes are so small and there's so many of them, rather than describing the pure absolute number of solutes, we describe them as the number of moles that are present. And moles is just a way of collect a big number that accounts for a large number of solutes at one time. For example, 1 mole equals 6.022 times 10 to the 23rd molecules. So it's a massive number. And so if you say that you have 1 mole of sucrose, then you have 1 mole of sucrose equals 6.022 times 10 to the 23rd molecules of sucrose. So a massive number of amount of sucrose. And so we will be using moles when we're talking about water potential here. Now, R is just the pressure constant. And so for all of our calculations, it's going to be the same number, 0.0831. It's just a constant associated for pressure. So R will always equal 0.0831. Now, T in the equation is temperature, and it's temperature in Kelvin. So you have Fahrenheit, Celsius, and Kelvin. We don't use Kelvin very much. It's used in different fields of science, but this equation does use Kelvin. And so you'll often be given the temperature in Celsius, and you'll have to know how to convert it. So to go from Celsius to Kelvin, you just take whatever your Celsius value is and add 273.15. So if you're at 0 degrees Celsius, you're at 273.15 degrees Kelvin. If you're at 10 degrees Celsius, you're at 283.15 degrees Kelvin, and so on. And again, I will give you that equation. So I, the ionization constant, C is the molar concentration, R is a constant, and T is a temperature in Kelvin. So it's not too bad. So let's go through a practice problem. So a gardener is trying to adjust the water uptake of her plants by modifying the concentration of a nutrient solution in which the plants are grown hydroponically. So the plant is in a container of water, and the gardener wants to ensure that the plant's getting enough water. And so she's going to adjust the nutrient concentration to ensure water is moving into the plant. She knows that the root cells of her plants contain a solution that has a solute concentration of 0.03 moles. The temperature in the greenhouse where the plants are grown is 20 degrees Celsius. So we know the molarity, we know C, we know the temperature is 20 degrees C, so 293.15 degrees Kelvin. Now, the ionization constant here, we're going to assume is 1 because they don't tell us what the actual solutes are. And then we know the pressure constant. So to get to the solute potential, I is 1, C, the molarity is 0.3, R is the constant, 0.0831, T is 20 plus 273.15, so 293.15. You put all that together, so negative 1, because there's a negative symbol in the front, times 0.03, times 0.0831, times 293.15, equals negative 0.731 millipascals. So it's not too bad. That gives you the solute potential, right? Just using that equation there. So next we're going to talk about pressure potential, also known as turgor potential, because it's talking about turgor pressure. Remember that if you put a bunch of water in the cell, it's going to start to expand it. It's going to put pressure onto the cell wall. That's turgor pressure. So pressure can be negative or positive. It can go either way. It depends on the specifics of the system. So higher pressure creates higher pressure potential. So the more pressure you have, the higher the pressure potential you have. And the way to kind of think about that is think about water will move from high to low concentrations, right? If you fill up a water balloon with water, you're increasing the pressure potential of the balloon because the balloon's kind of elastic, so it's squeezing the balloon a little bit, right? And if it's squeezing the balloon, it's harder and harder for water to get in. And if you stop putting water in and just kind of let go of the balloon, water is going to squirt out. That's because the pressure potential in the balloon is higher than it is outside the balloon. And so as you increase the pressure, the water potential goes up. Now, if the pressure potential is greater than 0, it's compression. So it's being squeezed, it's being compressed. If it's less than 0, it's under tension. It's starting to pull water into it. It's experiencing negative pressure, and so it's pulling water in. Imagine you had some sort of like a container that you can compress and it has one opening on it, right? And if you pull that container open, it'll draw air in. That's tension. That's negative pressure. It's also the way our lungs work. When our diaphragm extends, our lungs cavity swells up, it opens up, and that negative pressure draws air into our lungs. Now, plants can manipulate the pressure potential, and they do it by manipulating the solute potential. So remember solute potential here. You add in a bunch of solutes, the solute potential will go down, and water will move from high to low concentrations, so it'll move over to the lower water potential. Now, imagine you have a cell, right? It's enclosed in a cell wall and a cell membrane. If you throw in a bunch of solutes, like the plant pumps in a bunch of solutes into it, the solute potential is going to go down, right? And if the solute potential goes down, just like we see in this diagram, water is going to move into the cell, just like a hypotonic situation. So you pump in a bunch of solutes, water moves into the cell, and as water moves into the cell starts to expand. Now that expansion is just like the water balloon. So as you expand, the pressure potential goes up. So by adding solutes into the cell, the plant is drawing in water and increasing the pressure potential. This is why I was saying changing one factor may or may not change the overall total water potential, because one change can affect a change in the other parameter, like solute potential affecting pressure potential. Now, pressure potential becomes very important when we're talking about how plants draw water up the tree or the plant. So transpiration is evaporation of water from the leaf. So you have the leaf, water is going to evaporate out of the stomata into the air, right? Now, when it does that, it's drawing water out of the cells of the leaf. And just like how we just talked about, when you add water to it, the pressure potential goes up. Well, when water evaporates out of it, pressure potential goes down. And when pressure potential goes down, total water potential goes down. Now, remember, water will move from high to low potentials, right? And so if the leaf water potential is going down, it's likely that the water potential in the petiole, the stem connecting to the leaf blade, is still high. And so water will move from the petiole into the leaf. Now, this is really important when you're talking about, we'll talk a lot more about it later on, how water moves to the xylem. Remember, water molecules will stick to each other and other things. And so if water is moving from the petiole into the leaf, it's not just going to leave a big air bubble, although that is possible, but it's not likely. It's going to pull up a chain of water molecules into the petiole. And so this transpiration from the leaf, the movement from the petiole into the leaf, and the attraction of the water molecules creating a chain pulls water up the plant. Now, this doesn't occur, though, if the plant's in a low water environment. So wilting will occur if the water potential is low outside the cells, right? And so imagine you're a plant inside of a flower pot or something that is really, really dry. In that case, the water potential is actually higher inside the plant than outside the plant, and it'll start drawing water out of the plant into the soil. And then when there's no more pressure, there's no more water moving through the plant can't create trigger pressure. The plants start to wilt and they fade away. Now, when water potential is high outside the cells, water will move into the cells, just like we've been talking about, and create trigger pressure and stop the plant from wilting. So if you see a wilted plant, you know that the water potential in that plant was higher than what it was in the other area, and therefore it started to wilt because water started getting drawn away. So next is gravitational potential. It's always negative, and at ground level, it's 0. So as you go up, it becomes more negative. Now, the downside to gravitational potential, particularly if you're a tall plant, if you're like a meter tall, it's not going to really matter that much. But if you're like a giant coastal redwood, then you are experiencing all of this gravity pulling your water back down towards the soil. And so it's a big problem for big plants, but a negligible problem for small plants. Now, just like I was talking about where water molecules are going to get pulled up through the xylem, the gravitational potential will be overcome by cohesion and tension of water. And we'll talk about that in another video. We then also have matrix potential. So matrix potential is very similar to solute potential. Remember, solute potential is water molecules binding to solutes, and therefore those hydrogen bonds are taking energy out of the system and lowering the solute potential. The same thing is true for matrix potential, except rather than binding to the solutes, it's binding to other stuff, particularly the cell wall. And so it's binding to other things that are taking up that energy from water. Things that are hydrophilic, so things that like water, like the cell wall does. And so it takes up some of the energy of the system by just using up some of those bonds in other objects. Now, in a dry seed, it can be very strong and negative, and in dry soils, it can be very strong and negative. And the reason for that is there's not much water there. And so if you get a little bit of water, it's going to attract the water really tightly, really quickly. But as soon as you add a lot of water, it becomes 0. It doesn't really matter anymore because there's so much water in the system that it's negligible. Okay, so when we're talking about water potential, the main goal, the purpose we're talking about all of this is so that way we can understand how water goes from the soil to the root, through the xylem, up to the leaf, and transpires out. So for plants to work, for plants to function, the water potential must follow a predictable gradient. So it must decrease from the soil to the root. So the root must be less than the soil in water potential. The stem must be less than the root. The leaf must be less than the stem. And the air must be less than the leaf. If these break, then the plant will die. Now, if we look down here, we have the soil and the roots here. The root cells have a typical water potential of about negative 0.2 millipascals. The stem has one about 0.6, negative 0.6. The leaf has one about negative 1.5, and the atmosphere has about negative 100. So the root cells, water will move from the root cells to the stem because the stem's lower. The stem to the leaf tip, water will move to the leaf tip because the leaf tip's lower. And then the atmosphere is usually dramatically lower than the leaf. And so negative 1.5 to negative 100 is a huge difference. And so water will move from the leaf to the atmosphere. That's why water potential is important. It allows the water to be drawn up. Now, during this part here, the cohesion and adhesion of water molecules is going to be important. So water molecules sticking together, pulling up like a chain all the way up the tree. Okay, so we're just going to go through a practice problem here. And there's going to be practice problems in Canvas as well. But the goal here is to determine whether or not water will move into the root. So we have root cell measurements and soil measurements. The root cell, the solute potential we don't know, but we do know that it's at 25 C, so 298 Kelvin, and it has a molarity of 0.1. The pressure potential is 0.3, and gravity and matrix potential are negligible, so 0. Now for soil, there's no trigger pressure, so pressure, gravity, and matrix potential are all negligible and 0. The solutes are at the same temperature, so 25 C, 298 Kelvin, and 0.01 molarity. So here's the equation at the top. So solute plus pressure plus gravity plus matrix. And down here is going to be our calculations. So we're first going to calculate solute potential for the root cell. So that should say negative 1. Let me edit that real fast. So solute potential, -1, because we're going to assume an ionization constant of 1 because it didn't tell us anything otherwise. It has a molarity of 0.1 here. The R, the constant, you can see the equation down here. So R is the pressure constant, 0.0831 times the temperature in Kelvin, 298. That gives us a solute potential of -2.48. Now over here, we have this similar equation, -1 times for the ionization constant, 0.01 for the molarity, R is 0.0831 for the constant, and Kelvin is 298. It gives us a solute potential of -0.248. Now, when we add the totals up, we have 0.3 for pressure right here, -2.48 for solute, 0 for gravity, and 0 for matric. It gives us -0.278 for the total water potential. Now, if we look over here, pressure is 0, gravity 0, and matric 0. So our solute potential is equal to our solute potential total, or our water potential total. So our water potential total is -0.248. Now, -0.248 is bigger than -2.78. So water here will move from the soil into the root cell, what the plant wants. Water is going to move from the soil into the root. So if you can do problems like this, you should be very good for this part of the exams and quizzes. I'm going to put a lot of the assignments geared towards these kind of problems so you can practice that. So to recap, we talked about transport, simple diffusion, facilitated diffusion, active transport, and we talked about water potential, each piece of it, so solute, potential, gravity, and matric, and how to use the equations to calculate those. And again, remember, I will provide you the equations on any problems.